So welcome everybody to our latest Q&A from the Science for ME forum. Today's Q&A is with Dr. Mike, can I say?
Sure.
Yeah, Mike VanElzakker. I think that this will be quite an interesting, or more interesting discussion, as the topic that Mike looks at is basically the brain, which is pretty much the most complicated thing that you can look at. I think it's safe to say. So welcome, Mike. Thank you for your time.
Thanks.
And
thanks for inviting me.
I was wondering if you could just give us a brief background, of yourself, of your research, how you came into the ME field.
Sure. Yeah. So I'm a neuroscientist. Most of my training involves sort of the stress field and memory. That's sort of my background. And I still work in post-traumatic stress disorder, which is sort of my other life. Essentially, I got into working on ME. We'll call it chronic fatigue syndrome. The way a lot of people do, which is a sick friend. A friend that I've known for, since I was young, a kid. It's one of those things where when you reconnect with an old friend through the internet, you sort of, oh, what have you been up to? What have you been up to? And I say, oh, I'm, whatever, in Colorado doing neuroscience research. And she said, well, I had to drop out of, early acceptance law school. I got real sick and suddenly couldn't make it up the stairs. And this was back when, the term yuppie flu was being thrown around and things like that. And, just sort of being a curious person, I wondered about this. And also knowing my friend who is, sort of not a malingerer. I do think there's a slight cultural where it's somehow, you have to sort of preface by saying, oh, they were really hardworking and they were motivated and they were such a good student. And if they were at home on the couch, they would also deserve medical attention. But she was one of those that was hardworking and motivated and all that. And I sort of knew that it's unlikely that she would just sort of hit the pause button on her life for reasons of attention and that sort of thing. So I started looking into it and started thinking about it more, essentially, is how I got into it.
Okay, brilliant. So as I've already touched on, you're essentially, I think I'll be right in saying that you're looking for the clues as to what ME, chronic fatigue syndrome, again, whatever we want to call it, you're looking at the brain for that. Is that correct?
Yeah. So, right, exactly. We're looking for brain correlates. And, I am not under the illusion that with the current techniques, non-invasively, that we can find out everything that's going on in the brain, but we can at least find correlates and try to understand the mechanisms, perhaps, of what's, what's being driven in causing these symptoms. Just to be fair, we were joking a little bit about the CFS ME name just because that's a controversial topic, but I do understand why that's an important distinction to make. And it is a little bit different US versus UK, where here in the US, if you tell a doctor that you have chronic fatigue syndrome, at least they will know what you mean. And if you tell them that you have myalgic encephalomyelitis, they will have no idea. And they will say encephalomyelitis, what do you mean by that? You have an active herpes infection? Let's get you into the ER. You could be dead in 2 days. So I do use the word chronic fatigue syndrome here in the US, especially when I'm doing sort of public outreach with, medical people to try and help them understand. But anyways, I've segued a little bit. Yes, we look in the brain and we try to understand sort of the mechanisms that may be driving these symptoms, understanding that our current tools probably aren't going to let us get to the root cause.
I suppose my first question is, what tools do you use to look at the brain?
Yeah, several. I mean, most directly, we use MRI, including fMRI, which is just essentially a really powerful magnet that, everybody knows what an MRI is. You can get that an MRI of your sprained ankle or something like that to get a structural image. The F of fMRI is functional, and that it relates to the fact that, for example, if someone's anemic, they take iron supplements, right? So iron is magnetic. So hemoglobin that is bound with iron has slightly different magnetic properties than hemoglobin that is not bound with iron. And with a very powerful magnet, you can tell the difference between hemoglobin that is bound by iron and that is not bound by iron. And so we can tell where in the brain is being perfused by blood, essentially using fMRI. And then related to that, there's a lot of different techniques within MRI that we can use, like arterial spin labeling, where you essentially get a pulse of magnetization at the neck and you follow blood as it flows up into the brain. We use magnetic spectroscopy, in which in the same way that light spectroscopy, different chemicals, if you shine a light through different chemicals, you get a different spectrum of light. Same thing with magnetism. If you use a strong magnet on different chemicals, they will give a different magnetic spectra. And so we can tell some different metabolites that are common to the brain, the relative concentrations. Diffusion, which is a type of MRI where we can tell, because water molecules normally sort of move randomly, but if they're in, say, like a pipe, they don't move completely randomly, they move along the pipe. Well, it's the same thing with axon tracts, which is the long, thin part of the nerve cell. These water molecules move non-randomly down axon tracts. And so we can tell the thickness and shape of these axon tracts using diffusion tensor imaging. There's a whole bunch of things we can do with MRI. And then separately, there's a technique called PET, positron emission tomography, in which you take some biologically meaningful substance and you tag it with a small amount of radiation. When I say small, like if I was to fly from Boston here to Copenhagen, I would be exposed to about the same amount of radiation. It's a pretty small amount of radiation. Nonetheless, it's got a fast half-life, so it's decaying rapidly. So you take this biologically meaningful, radioactively tagged substance, inject it into the body, and wherever the radiation is decaying, you can tell that that's where that bioactive substance has been taken up. So it could be, for example, fluorodeoxyglucose, where we can tell where in the brain glucose is being used. You can make something that binds to receptor systems. What we use and what Nakatomi used in 2014 is a PET radioligand that binds to a protein called the translocator protein, which is a protein that's produced by glia, the innate immune cells of the brain, when they become activated. And so this is a proxy for neuroinflammation that we can use with PET. And then we can also use techniques like EEG, where we can sort of look at the very surface of the skull, we can see a small amount of electrical activity and make inferences about that. So there's a bunch of different techniques that we use.
Okay. So they're the tools that you can use. Have you so far used all of them, or just?
Yeah, we're just now kind of in talks to get an EEG study up and going. So we haven't actually used that on patients yet, but everything else we've actually used on patients at this point.
Okay. And can you give us a broad view of what you found, or what you believe that you found so far?
Yeah, so I think the interesting and most novel thing that we've done so far, we have a bunch of studies that are sort of up and running, and they're in the middle of, we're sort of in the middle of gathering data. So we don't want to make too much of a story out of anything, of course. But one of the things that we see is what we do in the scanner is we take a lot of peripheral measures of physiology, right? So heart rate, blood pressure, pulse ox, inhaled gas exchange, respiration in and out. That ought to correlate very well with what's happening in the brain, right? It's sort of the same system. And one of the ways that you can tell if there's problems with brain perfusion is to see the level to which those peripheral measures correlate with that bold oxygen response that I talked about, that the magnets can measure in the brain. And so far we've seen in patients much more than in healthy controls, there's a problem with that correlation. It doesn't seem like the peripheral physiology correlates as well as it ought to with the central blood flow. And you can make a bunch of arguments about why that is. But that's one of the things I think that's pretty interesting. And we've got preliminary data from a bunch of different measures, of course. We could talk about that, but I do think we have to be a little bit careful with preliminary data to not run off. I know it's a very eager patient community, totally understandable. But I think sometimes we grab onto every little bit of signal and blow it up. So we have to be a little careful with that.
Yeah, certainly we wouldn't want, like you say, we are a community, not just the forum, but wider, in a wider sense. Quite possibly because we do feel left to ourselves to a large degree.
You guys do a huge amount of the detective work that ought to be being done by medicine.
Well, thank you for saying so, but equally that does tend to translate into the merest hint of a sliver of a clue might then go on to be put forward as the big thing.
Sure. Which I get. The alternative extreme is, okay, go home and wait for a randomized clinical trial for that one drug that's going to just make it all go away, which is absurd. So I understand.
Yes, we very much are keen to hear any news, but at the same time, whenever we can, at least on the forum, we try and take a reasoned view of it. How does that fit with other studies, or even how plausible it might be or might not be. So from what you see and what you've seen and what you might theorize, obviously the big thing that really concerns all patients is post-exertional malaise, which really doesn't seem that there's some hints that either post-exertional malaise or something similar-ish can be seen in other conditions, but not many. Do you have any idea how that might be coming about? I mean, you talked about what you see from the brain not correlating to broader bodily functions, if I'm putting that right.
Yeah.
Would you think that would be some part of perhaps post-exertional malaise?
I would imagine so. I mean, if it's part of the baseline symptoms, then I would imagine it would be part of symptom provocation. I mean, one of the interesting things is that the study that I described is a PEM study where we do a baseline scan, a whole bunch of autonomic testing in the scanner at baseline symptoms. And then they go off to do an invasive cardiopulmonary exercise test with Dr. David Systrom, which we predict is going to make them crash. And we sort of take advantage of, hopefully in the best possible sense of that phrase, we take advantage of the post-exertional malaise, the expected post-exertional malaise, and we bring them in, didn't do the exact same scan sequence again, so that we can pick up the delta, try and understand what the difference between the baseline symptoms and the provoked symptoms are. One of the interesting things that we found is that it is not the case, again, preliminary small n, but it is not the case in what we see so far that that mechanism of failure of correlation has worsened during the post-exertional state, which, I mean, it's sort of interesting. So, and by the way, that is a definition of PEM, that is aerobic exercise, close to peak. So that's different than feeling sort of like wiped out after whatever, trying to read a newspaper or something like that. Right? It's a pretty sledgehammer provocation, but to my mind, just because something hasn't worsened with symptoms doesn't mean it can't be the driver of symptoms, right? I mean, if it's a failure of perfusion, then it's possible. Again, I don't want to run off on it, but it's possible that a failure of proper perfusion during exercise may have longer-term consequences that may take a little time to kick in. So it's possible that that's what PEM is. I mean, I guess PEM is a really interesting topic, and my understanding is that the way it's used now is not quite exactly the way that Melvin Ramsey used the term, where he talked about it, where the term malaise actually made a lot more sense. He talked about this sort of like bad kind of gross feeling that after people exerted themselves, it was that really was I don't want to say centered on muscles, but it really involved muscles a lot more than the way people talk about it, in my experience. Where the way people talk about it now is a little bit more like all symptoms exacerbate, brain fog exacerbates, and that sort of thing. So just an interesting sort of distinction there to keep in mind, I guess. And the question of what might be driving it, when someone, again, that sort of matters how we define the term, right? Because I do think that there's a likely different mechanism between whatever pedaling real hard at 60 hertz on a bike as the resistance on the bike is being increased by 15% a minute versus whatever, reading a book or watching TV and finding yourself sort of like mentally drained and your eyes are tired and that sort of thing. I'm not quite sure it's the same exact mechanisms, even if it may share sort of a similar endpoint. So for what it's worth, I do think we have to be a little mindful of that.
Sure. Certainly, there's been some discussion on the forum about essentially what a descriptive model of ME might be, and to put it in basic terms, obviously we which might link into you seeing no change from pre-exercise to post-exercise in the brain.
With that measure, I think that's really key, right? I don't mean in the brain per se, just with that particular measure.
Yeah. I mean, obviously for the vast majority of us, we can still produce a certain amount of energy. So there is that system which, while not working as well as for healthy people, it still works to an extent. But it's then at a certain point, which tends to be picked up by, an overexertion, particularly in physical activity, but not always, which then kicks us into, or would seem to kick us into what is described as PEM. So,
I mean, I could talk a little bit more about mechanisms. It is a science forum, so maybe you guys will be interested in. So, the way that I think about it with our model again, with sort of an aerobic, relatively severe challenge. I mean, it's in the scheme of things, it's a moderate exercise challenge, but we understand that it's provocative. So essentially, I think there's two plausible models from the way that we think about it. So skeletal muscle, we all know about cytokines, right? So these blood markers of inflammation, right? So there's a cytokine called IL-6 that is also considered a myokine because it's produced by skeletal muscle. So, when anybody exercises, they produce interleukin-6, which is sort of a relatively apex cytokine that sort of triggers a bunch of pathways from there. Importantly, and I'm not making a deconditioning argument, just hear me on this. There are distinctions between very fit people and less fit people for the amount of IL-6 that is produced by a given level of exertion, right? So if someone is relatively deconditioned, their muscles are going to produce a little bit more IL-6 than a marathon runner, right? Mm-hmm. All things being equal. And from our perspective, if we've got a sensitized afferent signal for detection of cytokines, which can happen after multiple infectious hits, there are certain, essentially if there's a pathogen or something, that system, which, is part of which is the afferent vagus system, vagus nerve, which functions as sort of a diffuse sensory organ for the brain, detecting inflammation. So a slightly increased amount of IL-6 touching a slightly sensitized system might cause an increased afferent signal, which leads to glial activation on the brain side of the blood-brain barrier. Now, I understand that there's some studies showing no differences in IL-6, but to my mind, sometimes those studies aren't super well controlled. It's a very, very messy dependent variable, which is one of the things that we tried to argue in our last paper. You sort of have to control an absurd number of variables, and you can't control the sensitivity of your receptor system to a given amount of IL-6. So essentially the IL-6 to brain glia is one plausible mechanism for that kind of PEM. And another is if there's a failure of perfusion, you know, you're not getting the appropriate amount of oxygen perfusion in brain, that's going to cause a sort of a low level ischemic response, where essentially there could be longer term, you know, a couple of days, couple of weeks consequences. Again, we think glial activation on the brain side of the blood-brain barrier is going to respond to that, like, you know, slight failure of perfusion. And those glia activate to a greater extent on specific circuits that I think, you know, possibly might underlie the symptoms for some people. And they're certainly going to disrupt the sort of signal to noise processing power of the brain, which is going to lead to brain fog and sort of difficulty concentrating. You know, to my mind, sensory sensitivity, that makes sense if you've got glial activation in the thalamus especially. So those are sort of two plausible models for how our challenge might trigger subjective PEM in the brain.
Okay, so to drag myself back to the many questions that I've got, which sorry for our viewers and particularly the forum members who suggested all of the questions, I'm not going to get through them. But one, there's a number of questions to do with glutamate and glutamine, and the reason that I bring this up now, because you're talking about sensitivity of various areas. One question is asking how or whether glutamate can cause glial activation, whether that would be part of what you've just been talking about, you know, as a possibility.
So right. So we, you know, we reported using magnetic resonance spectroscopy a relatively increased glutamate in brain with the post-exertional malaise, which is, again, preliminary. I wouldn't run too far with that. But glutamate, from my mind, is the more important story might be as an effect of glial activation. So part of what glia do when they activate is that they produce a bunch of neuroexcitatory modulators. So they turn on neighboring neurons ostensibly to try and keep them alive. But what ends up happening is it sort of wrecks the signal to noise processing power of those neurons. But one of the things that glia produce, along with ATP, prostaglandins, bradykinin, nitric oxide, cytokines, is glutamate itself. So activated microglia produce glutamate. And this is the reason, for example, that, you know, 10% of severe brain injuries end up with seizures, for example. It ends up being sort of a glutamate storm where glia turn on and won't turn off. So I would think of glutamate as maybe being a little bit more of a evidence of glial activation.
Okay, right. But that's interesting because Dr. Carl Morton from Oxford, one of our forum members, points out that in a talk he did last year, he presented, again, preliminary findings. So yeah. We don't want to make too much of it, but in the patients that he investigated, he found an increase in glutamate. So
yeah, I guess I thought it was 6 times the glutamic acid and sort of 0.6 times the glutamine or something like that.
Yeah, I don't have the exact details, but our member reports a 6-fold increase in definitely in glutamate. So
yeah, yeah, I think it's glutamic acid. I'll have to check and see that again. But that I think that was taken from blood,
Yeah.
which I think it is an important distinction there, because there's far more glutamate in the brain than there is in blood, and there's sort of active transport at the blood-brain barrier on the side that goes into the blood to transport glutamate that way. But there's actually protective mechanisms to keep the glutamate from blood going into brain. So there is a sort of a distinction. I mean, it's not like it's not meaningful. There are receptors in periphery for glutamate that would signal the central nervous system. But what we're doing is a little bit different, and they are somewhat distinct compartments. I don't want to exaggerate, of course. It's not like the blood-brain barrier is Tupperware or something, but there is a difference between blood and brain, which, by the way, I really think is an underconsidered part of why this condition, people say, oh, geez, we did all the testing and nothing. Well, yeah, you tested it in blood. And if the condition is happening in the central nervous system, there really, really is a ton of evidence, if you do your homework, that the blood and brain have different milieus. It's different. Of course they communicate. Of course they can, whatever, cross-contaminate. Things can diffuse. Things can be actively transported, but you can't make an argument about what's happening in the blood-brain barrier by looking at peripheral blood without being really careful.
Okay, great. Certainly, I think that answers quite a number of our members' glutamate and glutamine questions there. One interesting one is that we occasionally do polls on the forum, just non-scientifically to poll, what our members think and what they report. And a high proportion of our forum members, which admittedly will tend towards the more seriously affected, report tinnitus.
Yeah.
So is that something that could come potentially or theoretically come from glial activation? Is that a knock-on effect? Yeah,
I mean, that's a reasonable fit. There's a couple different sort of models for tinnitus, and I think you can move from way out, sort of, like sensory ear hairs, to way in. And I think you can get a problem at sort of anywhere along that pathway. If someone's been to a lot of concerts and they have tinnitus, it's a little bit different than if someone's got, possibly a neuroinflammatory condition and they've got it. But yes, I mean, glial activation is part of the story. And to my mind, I think it's probably related to some of the sensory sensitivity. And this is an EEG study that's kind of on my mind. I just had a meeting about it yesterday, and I don't know, it's very preliminary, but I'm hoping that we can look at sensory sensitivity a little bit.
Okay, excellent. So to backtrack on my list of questions, you recently posted a poster presentation that I think, was it one of your students?
Yeah, I've done a couple lately, but if it's the one that Paulita did, there's an annual event here called Clinical Research Day, where people, MGH is a teaching and research hospital, and lots of research happens here.
MGH?
Massachusetts General Hospital. That's where I work. Yeah. Where, every year people from all over different labs can present their research. And so it's kind of a day where, I can go chat with people from, dermatology, and, we just sort of all share. And so we presented the preliminary findings. We didn't talk about it in terms of a PEM study. We only presented the baseline data. But in terms of that perfusion story, the cerebral vascular reactivity. And, I mean, we get good interest, I think. People are interested. They want to know. I am extraordinarily privileged here to work with really good people, people that take it seriously, and they just kind of want to know. Once in a while you'll get a slight amount of like, what is that? You get a little question about, is it a personality disorder or something? But most of the time I think people are well past that here, at least. And so, yeah, I mean, it was I thought it was a great chance to present some data.
Brilliant. And in that particular one, I understand the challenge that you were asking patients to undertake was a breath holding one.
Yes. So yeah, in the scanner, we do several autonomic challenges, and they're sort of mild, but they're enough to sort of perturb the system. So one is we simply ask patients to hold their breath for 30 seconds, just breathe normally. And then when this light comes on, just stop your breathing wherever it is and hold. That'll get a buildup of CO2 in your blood, which causes vascular changes, and you can actually measure the extent of vascular changes in the brain by looking at perfusion. And again, it ought to correlate, the response to that, the recovery and response to that ought to correlate with peripheral measures. And we see that in patients relative to controls, that really gets disrupted. So we think that's pretty interesting, and we're hoping that we can, I mean, whatever, well, the data are what they are, but we're hoping that perhaps that trend will stay as we look at more and more patients. Had another scan late last night. So, yeah.
Okay, brilliant. Just to throw what might be a quick question and answer in, one of our members wonders if you see any value in vagus nerve stimulation. Do you think that could be beneficial?
Yeah, with the obvious disclaimer, I'm not giving medical advice,
Sure.
Talk to your doctor and that sort of thing. I don't want people on the forum to start a new thread about how to hook up your car battery to your ear and things like that. But yes. Yeah, I mean, I do. Vagus nerve stimulation is it's non-invasive, it's relatively low impact. You can do a very low dose at first. And essentially, there's a branch of the vagus nerve called the auricular branch that comes way up close to the ear hole, essentially here, like under the symbicontia right here. And if you send pulses across the skin there, that's enough to trigger an action potential down into the nucleus of the solitary tract, which then permeates. And, I've of course talked to patients that have tried this. It didn't do anything at all for some. It helped for some. Some people, it gave them migraines. It's not like vagus is the only thing happening here, right? The trigeminal is right there in sort of 3 branches. And if you don't properly, really, really, really scrub out your ear, you're going to get the electrical signal sort of moving across your skin, which is going to activate trigeminal, which might trigger migraine. So this is one of the reasons that I hope that people don't just sort of run off and do this ad hoc on themselves. But yes, I mean, for one thing, the efferent motor vagus nerve is essentially an anti-inflammatory reflex. So there's reasons why, it's not the case that when you're 2 years old, something causes systemic inflammation and then you're just producing inflammation for the rest of your life, right? There's a feedback mechanism. And the motor vagus nerve is part of what does that. So there's a synapse in the spleen that then produces anti-inflammatory cytokines. And so if part of the problem with this condition is inflammatory, I personally don't think sterile inflammation, if there is such a thing, is it. So it's not root cause. But if some of it is over signaling from inflammatory mediators, which I think there's reasonably good evidence for that, then that's one of the things that you could do. Again, you're not getting at root cause, which is less than ideal. But also you could possibly, in the same way that a pacemaker works, regulate an afferent or sensory signal. So, I mean, I think it's worth it. It would work symbiotically with mestinon, which is a acetylcholinesterase inhibitor that would allow more acetylcholine to be in the synapse, which is what kind of vagus uses. So I would love to do a clinical trial where we get a really tight cohort of people and sort of try a bunch of different interventions. Right now, I think we're at the basic research phase, but I think it's worth trying for people. Not giving medical advice.
Yeah, sure. Okay, thanks for that. So that was quite a focused question, making the questions a bit broader, either from what you see or just what you think about CFS, the name again. Do you see it as being one thing, or do you think that we are going to see that it eventually turns out to be a number of related conditions, such as has happened with other diseases?
Yeah, I mean, I think it's a really good question. I think we ought to start with the distinction between the entity of ME, I'll call it ME, and then the entity of the diagnosis of ME. So when someone walks up to me and says, I have ME, I don't know if they have quote unquote real ME
Hmm.
or if they have a lot of symptoms that overlap. And so practically speaking, I think a big part of our job is to, for lack of a better term, filter out those folks that don't have real ME and might have something that's treatable, something other than sort of real ME. Let's call real ME, you know, the outbreaks in the UK in the mid 80s, or maybe Akirari disease, or maybe what happened in Incline Village, assuming that those are all three the same thing, which I'm not convinced of. I'm not saying it's not, I'm just not convinced. I don't know. We haven't really properly investigated, to be totally clear. So, you know, a big part of it is, you know, I almost guarantee that there are going to be people watching this who don't have real ME, right? Who have something that looks very similar to it, and, you know, isn't the same thing that happened in the Royal Free Hospital or whatever, you know, pick an example. So practically speaking, you know, if somebody could give me a cohort that really was real ME, I would of course study it. But part of what we do is to try and build a pipeline to help find alternatives, and figure out why someone else might be dealing with these chronic symptoms that are so very similar. Um, yeah, so I mean, I do think that there is a sort of a real ME. Um, I personally think that it can be sort of driven by different things. I know that people disagree on that. And maybe some people think that there's sort of one virus or something that's driving it. That's back to Koch's postulate, hot theory from the 1800s, about how pathogens work. I think now we realize more that it's community dynamics. You know, it's pretty rare that a single pathogen sort of works alone. It can be sort of the dominant pathogen in a community, and sort of drive a dysbiosis and that sort of thing. I mean, I don't know the answer. I would like to get at that. And at the very least, I think we can investigate as though that were a plausible option, which I think it is. But as to whether it's just one thing or not, I don't think so. I think that by the time that you start filtering out and paring down, if you can get to the point where you go, oh, look it, this is a group of people that all got sick in the northern UK in the mid 80s, then yeah, okay, maybe that's one thing where there was one thing that had sort of long-term consequences. But when you can have something that's triggered by Giardia, that's triggered by Epstein-Barr, and, you know, that's triggered by some bacterial infection too. I mean, you know, I don't know, but I don't think so. It's possible that it just weakens the immune system for something other, you know, something else to move in and drive the symptoms. But I suspect it's going to be more than one thing.
I suppose, arguably, if you've got a common resulting pathway, but triggered by different um, uh, causes, it's almost splitting. Well, arguably, it's splitting hairs. You know, to say that it's one thing or it's multiple
Yeah.
things.
It's somewhat of an academic debate. I don't mean that in a way to minimize what people are dealing with, but, you know, if someone is standing in front of me with these symptoms, it's somewhat academic about whether it's real ME or not. I mean, I do think it's important to do our best to have clean cohorts. And, you know, one of the things, you know, the first half of the study we were doing, we did multiple criteria, you know, ICC criteria and all that. And we got this sort of like a somewhat messy result. And so we're going to try and tighten a little bit to see if that makes a difference. So we're doing all the criteria, and also working with an MD who doesn't use any of the criteria, but who has had experience for decades with patients, and is going to give us a thumbs up. Yes, I think this person has real ME. And this is a doctor who does, you know, serious, thoughtful, investigative work about alternatives. You know, I always say that when if someone has the diagnosis of MECFS, it means basically that a doctor somewhere said, I give up. And they may have said that after doing a really careful, thoughtful, thorough history and analysis, or maybe not. And part of what we need to do is to figure out what all we need to do that thoughtful, thorough analysis, to perhaps find some people in the community that have an issue that can be better addressed.
I was right. That was a broader answer.
Pretty broad question.
but
I mean, it's a important sort of theoretical framework that is part of the reason that the literature is so messy. I think it's a significant part of why the literature is so messy.
Well, certainly, I think speaking from our forum members, we're very much in support of tighter cohorts used, but also at the same time understanding that in clinical settings where there's no research involved, you can't apply the same criteria, or you can't necessarily apply the same criteria, because like you say, if somebody's turning up with a number of symptoms that match the broader whatever it's called, they need to be treated, rather than saying you don't tick every single box on this research criteria, please go away and sort yourself out.
It's a classic problem across research, is the internal versus external validity, and they're always going to be at tension with one another. So I mean, I do think that there would be enormous value in finding a group of people that all got sick at the same time, for example. You know, there are studies of Incline Village survivors. It's not true that they're not being studied. They are. So that sort of cohort is really valuable, for sure. And then I think it's also valuable to study people that have this endpoint of the criteria, and sort of trying to figure out how they ended up there.
Great. So we've been touching on the subject of essentially what is ME, what isn't ME, and a hot topic at the moment is CCI.
Yeah.
Which many people have views on, and I wondered, if you could give your opinion on where we are with that, whether there is value in discussing it, just your point of view, really.
Yeah. So obviously Jen Brea is a prominent patient, and we've talked, and she's okay with me talking about the case publicly. So, but I guess for the people that think perhaps that CCI is not sort of real ME, what I want to know is how many of them saw Jen's movie and Jen's case before she found out about the CCI, and from just viewing those things said that's not real ME. That's what I would like to find out, because the question, the really important question to me is that she saw almost all of the top ME doctors at least in the United States, probably elsewhere knowing Jen, and none of them flagged it as, wait a minute, this isn't real. I mean, she had idiosyncratic symptoms, but that's sort of the norm, right? So I think at least mechanistically, it ought to be interesting that, let's just say for argument's sake that it's not, okay, just for argument's sake purely, it's not real ME. The fact that this other thing fooled everybody into thinking that it was ought to be interesting mechanistically. So what is it about compression at this area of dorsal brainstem that led to symptoms that sure look like they overlapped with ME? And again, this gets to the question that we talked about before of the sort of real ME versus the broader diagnosis. When someone walks up to me and says, every time I stand up, my heartbeat goes crazy, I feel really wiped out after I exercise, et cetera, et cetera. I think CCI and these structural problems are part of the, for lack of a better term, filtration process that ought to go on as we're sort of digging and trying to find a problem. But, Byron Hyde talked about the idea that people with Ehlers-Danlos for some of the most severe patients. You know, I personally see it as this concept that I've talked about called equifinality, where if you've got a forest fire, you know, maybe it was from lightning, maybe it was from fireworks, maybe it was from a careless smoker, but you've got a forest fire now, and that's the endpoint that's shared. And perhaps it can be triggered or caused or driven by multiple causes. And I personally think that you know, I mean, unless you just don't believe Jen that she was sick before and is better now, then I think you can't argue that CCI wasn't driving those symptoms. So something about the brainstem being compressed at this area, from my perspective, it's where the nucleus of the solitary tract and dorsal motor vagus are. You know, I mean, she's got she was having these problems sort of late on, where you know, she had to sort of like continually breathe on purpose. You know, that's a function of nucleus ambiguus, which is right there. So these sort of brainstem nuclei that, to my mind, are really important in communicating inflammatory milieu between the inflammatory you know, immune system and the central nervous system, are being compressed, which is another way to activate glia. You can do it with pathogens, you can do it with damage, you can do it with compression, you know, a whole bunch of different ways. So that, from my mind, it's part of evidence that this general system is being constantly provoked. And that the question for each individual patient that walks up to an MD is, why is that happening?
So again, it could be one of the multiple triggers that we're theorizing, you know, could activate well, particularly in our discussion here, the glial to.
Yeah, for sure. And I do, you know, I do think that trigger is not a useless term, but I think a lot of the times I use the term driver because I'm suspicious of the idea that something, for example, a pathogen comes in and affects the immune system so profoundly that it's dysregulated for perhaps the rest of someone's life, certainly years, and yet that same immune system has defeated it. And, you know, I think there's not nearly enough, you know, sort of like microbiologists in this field, people that have expertise in pathogens. You know, we know, for example, that just as a single example, herpes viruses like to go latent. They like to go latent in nerve tissue. When they are latent in nerve tissue, even engineered so that they cannot activate, so they are pure latency, they cause an ongoing cytokine transcription in peripheral ganglia. So something that is, you know, it could still be driving this. And by the way, that's not going to be detectable from doing DNA and RNA assays of peripheral blood. It's not how it works. Unfortunately, it's a tough problem. But yes, I mean, I just to add a little nuance, I like the term driver in general.
Okay, great, thank you. Just a couple more, I think, as we're approaching our allotted time, more or less. Do you, given that we've talked quite a bit about activated glial, is that something that, obviously dependent on the tech at the moment, is that something that could be scanned for? Is that something that could be if it's shown to be in all true ME patients, is that something that could be scanned for in the future, potentially?
I mean, it can be scanned for now, but the question is, what discriminatory validity does it have? And the answer is not much. I mean, so for one thing, when we talk, you know, we use the term activated glia as a bit of a shorthand, described in a binary sense. It used to be historically, you know, M1 and M2 activational states. It's really sort of on off. We know now that that's just grossly oversimplified. You know, most of the research happens in mouse models, which we know human microglia are, you know, maybe, I don't even know, an order of magnitude more complex in their activational states. You know, as of a couple of years ago, there were, I think, 19 distinct activational states that we knew of. And, you know, we're right now able to tell kind of on off, like that's it. So it's possible that there are, you know, there's some activational phenotype that's particular to this condition or the symptoms of this condition, which I think would be really worth chasing down, to be honest, because, you know, it's not the case that you guys are just all having seizures all the time, right? Just constant grand mal seizures, right? So, you know, this is one possibility of glutamate and glial activation, right? So there has to be some nuanced state somewhere between extremes. And yes, I mean, I hope that we get beyond translocator protein presence or absence, which is very, very blunt. And you know, just to be clear, I've talked about this publicly before, but glial activation is in everything. It's after brain injury, it's in bipolar, it's in schizophrenia, it's in psych disorders, suicidality, you know, Parkinson's, Huntington's, you name it. It's a very broad measure of, you know, sort of something's not right. Or at the very least, we're trying to fix something or we're trying to address something. And so again, I keep talking about root cause, but getting to that root cause of what's driving it in each individual, I think is probably going to be really key. But as far as future brain scans, yeah, I mean, I hope so. But right now, it's just so blunt, it's so broad.
Okay. And finally, the wider MECFS research area, do you see, you know, funding, collaboration? Do you yourself see any changes there? Because we previously, well, like we said at the start, the patients can tend to grab onto minor details because we feel that we've been left on our own, and I suppose I'm fishing for more good news.
Yeah. I mean, you know, I do think that there's a culture of wanting to collaborate. You know, you can have gestures and proclamations, and I think what we need is a material change. And so what I personally would like to see is something similar to what the autism field does, where they've got essentially in every patient that does research that's been funded by NIH has essentially an anonymous ID. And if they're at a research study at Massachusetts General Hospital, somebody loads their data up on this anonymous ID and it's available to other researchers. And then if they go to Ohio State University and do another study with another technique, another method, they also load it up under that anonymous ID so that the data in a relatively raw state can be shared and correlated. I think this is really important for disorders where there's a longitudinal story, which I do think the story of ME, an important factor is how long someone has been sick. You know, I think that there's going to be, that's just a really important factor to take in mind. I mean, I guess what I would say is to have the material change of data sharing. I mean, there's people talk a lot about sharing data and collaborating and that sort of thing. And I mean, I've only been doing this for a little while, so I can't speak of what it used to be like, but I don't know. It could be better, I guess is what I'll say. And in terms of funding, yeah, of course. I mean, we could use more funding. Weirdly, I would like to spend a lot of money on research not on these patients. I would like to study mechanisms, basic mechanisms. The best evidence we have so far of neuroinflammation in this condition is probably Nakatomi 2014, arguably, right? So they found with the PK11195 radioligand elevated translocator protein. What does the translocator protein do? We don't know. We know that it's activated during microglia activation, which again, there's like 19 different states of microglia activation. Which one, you know, so just research at a basic level. What does this stuff do? How does it work? I think is going to be really beneficial to both this community and others. And this is why if we doubled NIH funding, then, you know, I think that is really important. I think that activists ought to be focusing on that instead of NIH, please reslice your tiny pie. We ought to be saying, look, a healthy society should double its research into these biomedical questions, because it's going to benefit us and it's going to benefit whatever, Alzheimer's, you name it, Huntington's, you know. So yeah, that's my little blurb about how to affect the funding thing. But I know NIH gets dragged a lot, but like, I kind of believe them when they say they don't get a lot of good applications, you know, like that's not mutually exclusive with the fact that they ought to fund more. But this is part of why I try to go to conferences for other fields to say sort of, hey, Alzheimer's people, think about this condition too. You know, people that are doing mouse models, when you talk about glial activation, list this condition too as the conditions that are relevant to what you're doing. So I just, you know, broadly speaking, we ought not be in a sort of scarcity mentality when we're doing research on chronic illnesses that affect people.
Well, certainly one thought that I've had is that the evidence seems to pretty strongly suggest that we have an incorrect response for us, overexertion. You know, that whole mechanism of that healthy people use to get fit.
Yeah, right, exactly. Yeah.
Or not even healthy people, other ill people, they use, they can improve their fitness. That mechanism just isn't working for us for whatever reason. And to my mind, that would suggest if we can figure out what is broken there, what part doesn't work, that not only will it help us, but it will provide insight into the broader mechanism,
yeah,
which could potentially then be applied to so many other areas, you know, that obesity potentially.
Sure. Yep. And that's, I think people have no idea how poorly that kind of stuff is understood. So if you put a running wheel into a rodent's cage in a lab, it will run for miles and miles. People don't realize that, like kilometers and kilometers, I suppose. But it is unnatural for these animals to be just sitting there sedentary in a cage. And that is 99% of the research is done on these artificially sedentary animals. And so it may be the case that we're missing a huge amount of just how these systems normally work under normal circumstances. I mean, yes, I mean, I agree that the exercise and exertion, just, you know, and this is basic research that could be done. Again, I am all for working on patients, absolutely. But at some point, the tools we're using to measure these things in patients are not up to task. And, you know, we're going to do another cytokine study and find, yes, it seems to be abnormal. We have no idea what that means. And, you know, I think getting at the mechanisms, I think are really important, and that can be done with basic research.
Okay, brilliant. I'm kind of out of questions. Well, we're out of time, and I'm pretty much out of questions, but the questions I've got would launch us into another big chunk of time. So
okay, for another time, I'd be happy to do it again.
Well, that'd be excellent, because
depending if I get dragged in the comments, if I get reports back from my lurker friends, maybe not, but no, I'm just kidding.
Well, no, I'm sure it rarely is anything black and white. So
that is very true.
So I think our forum members tend to have an understanding of that, although we do go off at times on particular subjects.
Okay.
But I'd like to think that our members will have that, at least an appreciation of the grey areas that tend to exist.
Well, just understand that I have an effective counter argument to whatever that person said.
But yes, we would love to revisit at some point, you know, especially, you know, if and when you have a new announcement of study results or whatever.
Or just to talk about mechanisms, to be honest, like I think that we ought to, weirdly, I think that we ought to sort of pull back from what's happening in patients to an extent and think about mechanisms. You know, like, yeah. I just think that that's the sort of thing, you know, how do you end up in these states? How do these systems work? Some basic science, I think, would be really fun to talk about too.
That'd be great. Perhaps we'll pencil it in then. So thank you very much. We'll stop this now.
Okay,
So thank you.
So much.
yeah, thank you for your time, and we'll look forward to speaking to you soon.
Okay, take care.